My name is Margaret Gruca, and this video is going to be an introduction to nascent transcription analysis. So nascent transcription analysis is defined as RNA that is currently being transcribed by the genome by one of the RNA polymerases. So it's kind of this intermediate step shown here by this green arrow, one of the two green arrows, in between DNA and stable, mature RNA. So steady state is the study of expression of DNA to RNA, and really examines the stable cellular RNA transcriptome. So this includes elements such as transfer RNA, tRNA, messenger RNA, microRNA, ribosomal RNA, etc. You get the point. Anything that's stable, and oftentimes is post-editing. So things like splicing, capping, poly-A, so on and so forth. So you can imagine we get different types of data between steady state and nascent. So steady state, which we're just going to use RNA-seq as an example in this video, you can gain information such as gene splicing, so you can get information on isoforms, different gene isoforms. You can get information on differential expression of steady-state RNAs, so you can imagine mRNA, gene-expressing transcripts, protein-expressing transcripts, post-transcriptional modifications. You can get information on exon and intron boundaries, so on and so forth. So basically anything post-transcriptional that includes some sort of editing. Nascent, on the other hand, so grow-seq, pro-seq, the big new element that we get through these types of analyses are these intergenic transcription regulatory elements, or TREs. It's kind of a mouthful, but it's basically anything that shows up as being transcribed that's not over an annotated region. So a lot of times we'll call these eRNAs if they're sources of bidirectional transcription, and they'll have a certain shape to them. We'll get to that in a little bit in the video, and how we go about identifying and naming these elements. You can also get RNA polymerase behavior out of nascent, right? So over an annotated gene region, we can see where RNA polymerase appears to be binding and loading. You can see the initiation peak, you can see where it goes into elongation, and where it eventually terminates. You can also get immediate changes in transcription upon permutation. So let's say you treat a certain cell with a drug. Really, it takes at least 40 minutes for the shortest gene to be transcribed and translated into protein. And so any data that you get that's under 40 minutes really is representative of the immediate effects of whatever that perturbation was, or whatever that drug was, whatever that hormone was. So you can see really, really short-term effects of these different perturbations. So here we're going to show you, this is similar to what you would see in IGV, right? So this top track here would be something like a bed file. In this case, it would be a bed file of gene annotations. Okay, so this red canonically represents the antisense, blue is sense. So each one of these little tracks here is a gene. Okay, or not tracks, but regions, annotated regions. Okay. So the second track here, this is showing an example of a nascent track. So you see that there appears to be a lot of transcription over what would be an annotated gene region here. You see, again, on the sense strand, still over an annotated region here, another antisense, another sense, so on and so forth. Notice that you get contiguous regions, really, of coverage, right? Again, since we're not just getting pre-editing, we're going to get everything that's over that gene, exon, intron, and everything in between. You can see down here we've got the bottom track, we've got steady-state RNA. So you see these kind of sharp peaks, and if we trace our mouse cursor up, these are over exons. So these little dashes, these little lines that are over the gene are the exons. So you can see there's a lot more intron to exon, but you get these peaks, and it can give you information on which isoform is being expressed. Okay. So a little bit different, and you can start to visualize the differences between these two types of analyses. The other element that I mentioned in the last slide that you can pick up using nascent is you can pick up these intergenic elements, right? These TREs, transcriptional regulatory elements. That's what we call them. If you knock them out, they appear to have some sort of effect on local transcription. Local is relative. It could be 25 kb, 50 kb. Just depends on how you define it and what you define as significant. However, you can see that, as I mentioned in the last slide too, you get this sense and antisense transcription, or what we tend to term bidirectionals. And we'll talk about these a little bit more later and how we can pick these up, identify them, and call them using different modeling programs. Okay. So the nascent pipeline is essentially the same as RNA-seq. So we're not going to talk a whole lot about it. Really, the main difference is that you do not need splice-aware alignment. So you could use bowtie 2, as is devised in previous years. Otherwise, HiSat2 works just fine. There's an option in HiSat2 where you can just turn off splice-aware alignment. The one thing that I will say is that there are a lot of different types of protocols for nascent, and as such, we get a lot of different kind of sequencing data coming off of whatever sequence we're using, Illumina, etc. So I will recommend that you use, that you really pay attention to whatever trimming program you're using. I tend to use something that's called bbduck, which is an extension of bbmaps. The reason that I do this is there are a lot of trimming options in this suite. So it allows you to really kind of fine-tune to the protocol if you know what you're looking for and if you, come up with weird kind of QC results. We will often do counting over annotated regions for downstream analysis to look at differential transcription rather than expression. However, again, all of the mapping pipeline is essentially the same with the exception of the splice-aware alignment. So what is a lot different is our downstream analysis. So really what we're trying to do in Nascent is we don't have annotations for things like eRNAs. So we're trying to find these using this annotation agnostic modeling. So what we do is we say, okay, an eRNA looks like, has positive and negative reads. It appears to make a certain shape. So how do we pick up and define these elements? So what we're really going to focus on here is the downstream analysis using programs such as Fstitch, Tfit, MDS, all of which were developed by our lab. So again, what novel elements are we really looking at? We're looking at things like these transcription regulatory elements, TREs, and this box in orange. If you zoom in on this a little bit more in IGV, this is actually bidirectional here. Just for a note too, top track is untreated P53, bottom track is P53 wild type treated with Nutlin, a potent activator of P53. May have talked about this already. You can see in this red box here too, this is, appears to be, this is over a gene, this is over a CDK1A, potently activated P53 gene. You get this kind of initiation peak. This isn't quite as clean as some others. But you also appear to get this kind of drop-off in transcription right around here. This might be a site of RNA pausing, which is another kind of hotly debated topic in the field. Somebody else in our building, Dylan Tages, he takes a look at this and tries to explain why this is happening. You can see this period of elongation here. Then you can see this 3' run-on in this blue box. So it appears to go past the end of the annotated gene. We don't really know why this is. We do know that RNA polymerase tends to run on further during stress. Okay. The other thing that we can get is information on Motif and Motif displacement. So in other words, if I say that this is my polymerase loading, how far are the motifs for the transcription factors away from this polymerase loading site? Okay. So we're going to talk about all these different elements. So the first one I want to introduce is really as simple as conceptually. It's basically saying what parts of the genome are on or off. So you can see this would be kind of an example, this figure here, example of a bed graph file. Where we've got a lot of reads over about a 300 base pair region. Fstitch would call this on. Okay. Fstitch uses a hidden Markov model and logistic regression to basically become a machine learning program, right? You can see on the other hand, this right-hand side, there's really only 2 reads interspersed over kind of a large region. So really, this is, this could be due to mapping error. It could just be random reads. RNA polymerase is kind of promiscuous. It binds all over and can, transcribe random little elements, even though they don't have any real effect on any sort of genomic transcription or gene transcription, I should say. And so really, we don't want to look at this because we can't really gain a lot of information out of this. We can't model it. So we just want to ignore that. Okay. So by ignoring that, we can use Fstitch as a pre-filter for things that are more computationally expensive. So something like Tfit, which we'll talk about in a minute. You can see if we plug in, we train some files to Fstitch. So in other words, we visually call on and off, feed our visual calls to Fstitch, and then Fstitch tries to learn what we would call as on and off. So this is an example. These bottom 2 tracks are also bed files. So you've got your one bed file, which is your gene annotations, and then you have these bottom 2. You can see that we've got a positive strand, negative strand, and that Fstitch is nicely calling this area of red. So this area of negative strand transcription as on, this area of positive strand transcription as on in the blue here. And you can see there's some kind of noise and some reads over here, but anything that's in green in Fstitch is getting called as off. So that's good. This doesn't really look like anything visually. It's nice that Fstitch is also picking it up the same way I would interpret it visually. That's good indication that it's working properly. And we're just going to ignore that and focus mainly on this region here for, say, modeling RNA polymerase activity. Okay. So the other thing that Fstitch can do is imagine if you give it another annotation file, you can start to inform yourself as to which regions of the genome are being actively transcribed. Are regions over the gene? Is it intergenic? Is it 25 kb upstream? Is it 50 kb upstream? Again, this could all be off annotation files, but Fstitch would nicely, fit those called regions on off over whatever regions of interest you have. So as I mentioned in the last slide, Fstitch is also often used as a pre-filter for Tfit. Tfit's primary purpose is to model RNA polymerase activity. So it models this initiation peak where polymerase loads. We can look at pausing ratio. We can look at this 3' run-on, which is something our lab is currently working on. We can look at things like these enhancers upstream. But Tfit has a ton of parameters. All right, so Tfit requires a bed graph or a coverage file, a bed file with annotated regions, such as the Fstitch output, and a config file that specifies all these different parameters for modeling, which can be unique to each experiment. So polymerase isn't always going to behave the same under each permutation and in each experiment. So mu here represents the typical loading position for polymerase. So let's say we have a peak here. Mu might be right in the middle. So that's where polymerase should typically load. Okay. We also have a certain amount of uncertainty, right, or variance associated with this polymerase loading position, or mu, okay, which is represented by this omega here. Okay. So from these two, we model polymerase loading as x, which is the actual modeled loading position for RNA polymerase. Furthermore, we can define a footprint. So over an initiation peak, typically they'll have pretty good positive and negative coverage, as you can see here in this bidirectional, or below it here. But in regions that are lowly transcribed, we may not be able to pick up this middle region quite so well. Okay, so we can incorporate a footprint, so especially over things like eRNAs, which are sometimes lowly transcribed, especially not under any sort of treatment. So we can plug this in and say there might be a gap between the positive and negative reads, so let's go ahead and include that anyway as a potential call. So could be very important, could be irrelevant, depending on how good your depth, coverage, complexity, etc. is, and depending on what elements you want to pick up. So that's why we make that modifiable. So there's also a rate parameter, lambda, and this is basically how quickly RNA polymerase II releases after loading, so initiation. So we take this signal peak here, mu, loading a polymerase with some variance, and then we say, okay, at some point it's going to release, it's going to kick off, as you can see down here, and it's going to go into this elongation, so this kind of big, long, however long, it depends, seems to depend a little bit on the gene. It seems to be relatively consistent between different cells. Okay, so Tfit, again, primary purpose is modeling RNA2 polymerase behavior. Secondary purpose would be picking up novel elements such as the eRNAs, so those bidirectionals that could be transcriptional regulatory elements. So lastly, I want to talk a little bit about MDS. So MDS computes the proportion of motifs falling within a given distance over an annotated region. So in other words, if we have an annotated eRNA, how many motifs fall within that region and also co-localize with the center of that region where RNA polymerase II loads, or mu, as given in the last case. Okay, so to make that a little simpler, let's say this is our annotated region down here, where I'm showing with my mouse. This is the origin, as called by Tfit, so that mu, or that eRNA origin, where RNA polymerase loaded. How far away are these different motifs? All right, so this would be one transcription factor motif, ATF3, June, SP1. SP1 is everywhere. But you can see that it's also falling kind of in the center of this bidirectional. Okay, so we can provide a bed file such as Tfit calls from max2, so from maybe chip data, ATAC, and it says which motifs fall within these annotated regions. Okay, we use Hokomoko database in this case to we use the position-specific scoring matrix, or these PSSMs, to give us information on canonical TF binding sites. These aren't always great, okay? And there's a big difference between Hokomoko version 10 and version 11, in that they cut out almost half of the annotated TF binding sites, all right? If you're not familiar with what a PSSM looks like, we included that here. So right here you can see if there's a letter bigger, so if there's a nucleotide, in this case G at this position, that's bigger, the TF prefers that over other bases. You can see in the position right next to it, C and T are about the same, so it'll kind of preferentially bind either one of those, but it won't bind A or G as strongly. Okay, so we do this over enough positions, and we say, okay, this is where this TF likes to bind, and so we use these, these PSSMs to determine basically where we predict TF binding. All right, so again, that's this lower right-hand figure. So in figure A, again, we've got this kind of eRNA origin, or mu, which is defined from our input file. And then over here, we can see something that we call barcode that we can get out of MDS, right? So each one of these lines, each blue line here, which is kind of faint in this top barcode, which is vehicle or DMSO, so it's our control, indicates a p53 binding event. In the vehicle or the untreated cells, there's very little p53 binding. It appears to be randomly distributed over a region, so over this 3 kb region. So upon treatment with Netlin, which is a potent activator of p53, as mentioned before, p53 strongly co-localizes to the center or origin of the eRNA, seen by this dark blue, so this dark blue kind of strip right here in the middle. And so which also happens to be located centrally over this annotated p53 motif. So you can imagine this is my origin, going back to figure A here. If I had a p53 motif right underneath it, all of a sudden I see a ton of motifs coinciding with this eRNA. Okay, so they're co-localizing together, whereas before I wouldn't see any sort of coordination between the two. So what does all this gain us? Okay, what's the point? So putting this all together, Fstitch, Tfit, MDS, and then even DE-seq, right? We saw DE-seq with RNA, RNA-seq analysis. We can gain information about how these transcription regulatory elements may significantly affect expression in differential treatment conditions. We say may because we have to obviously go through and experimentally prove this, but if we see these areas of transcription regulatory elements or proposed transcription regulatory elements changing with localized genes, co-localized genes, then it could have an impact on transcriptional regulation. And this has been demonstrated in past experiments. So this figure here that I included in the slide is a really nice example of all these tools coming together. You can see two very strong bidirectionals. You might say, wait, these bidirectionals are over an annotated gene down here in this track. However, this is an intergenic. So an intergenic enhancer is what we would call these. Or sorry, intragenic, intronic enhancer is what we would call these. So they are not over one of the, this would get typically spliced out. So we still call this as an intragenic enhancer. They don't typically fall over over exons. So you can see first two are examples of nascent data. And then this bottom track here at the top, this is actually chip data. Okay, so you see two really nice strong chip peaks over these bidirectionals, so over what we might call these enhancers. You can see in the bottom here, this first track underneath the annotated gene is Tfit calls. So Tfit is nicely calling both of these bidirectionals here. You can see Fstitch calls, a lot of this stuff is on. If we zoom in, we see a lot more transcription over this entire region. And then you can also see in the bottom here, this would be an example of MDS output, right? So it's saying there's a motif here and here that also nicely co-localize with these nice strong bidirectionals, chip peaks, and Tfit calls. So pretty cool example of all these tools working together. And again, if you imagine, you can couple this with something like DE-seq for differential, in this case, transcriptional analysis, to see if there are any nearby genes that appear to be affected if you were to, say, knock out one of these enhancers. So one of our future goals is to build a tool that'll take in all of these files, all these annotation data from things like Tfit, Max, MDS, and examine the effect on a gene within, say, X kb distance from given target genes. Okay, so if it's 5,000 base pairs away, is that significant? 25 kb, etc. So the last thing I just want to show you guys is give you a better picture of what Nascent adds to transcription analysis, right? So the top cluster of tracks here are actually CHIP. You can see our nice key down here in the bottom. Okay, so CHIP is going to bind a lot to intronic regions. Okay, it's going to bind other intergenic regions. Not a lot over exons. So this first little strip here, the color resolution may not be fantastic, but the first here is actually exons. Second cluster is nascent. Okay, and you can see you get a lot more over exons, which is all right. Should pick those up. You still get that intergenic. You get more over these 5' UTRs. That makes sense too. So a lot more kind of whole genome coverage. Bottom tracks here is RNA-seq, right? So you get tons of exons proportionally. This is all percentages, by the way. So this bottom track is percentage, not total number of reads. So this is a little misleading. But then you get a little bit less of the 5' UTR. You get a whole lot less of introns. That makes sense. Apparently our annotations are not perfect, or all of them. Some of the RNA may not be pre-spliced, etc. But really, like I said, nascent fills this gap, right? Gives us kind of a complete picture of what's going on in the middle. CHIP will tell us what's binding. RNA-seq will tell us what happens after transcription. Nascent tells us the in-between. Okay? So pretty neat stuff. Again, pipeline is very similar to RNA-seq, so you can go back and revisit that video. The only thing you do not need is a splice-aware aligner to get it mapped. After that, a lot of these tools are in development, and you just got to kind of play with them to see what's going to work best for your data. Thanks for listening. Hope you have a good time with the rest of the workshop.